Hello everyone. Today I'm going to take you to the world of microorganisms through the magnifying instruments called microscopes. I'll show you the difference in the pictures obtained using all these different types of microscopes. Microscope is a window to the invisible world of microbes. Our eyes can see 100 micrometers or microns without using any aid. Most bacteria are about 2 to 8 microns in length. The largest bacteria, called Margarita magnifica, is about 9,000 micrometers long, almost the size of human eyelash. In this picture, you can see the size of the bacteria and viruses in comparison with the human hair. What is magnification? Magnification is the process of enlarging the apparent size of an object. Its simplest example is a pocket magnifier. It is a convex lens that makes things look bigger. This is also called a simple microscope. Simple microscopes are commonly used by jewelers and watch mechanics. You can get a simple microscope called foldscope that has a single lens having 140 times magnification. When used with the zoom lens of a mobile phone, it can give magnifications up to 1000 times. You can see larger microbes under this microscope, including fungi, protozoa, and some bacteria. This is good for children who want to experiment with the microbes around them. Microbiologists, however, use the compound light microscopes in their labs. These microscopes have 2 or more lenses to achieve higher magnifications. First lens is called the objective lens. It is placed very near to the object that is to be magnified. The other lens is called the eyepiece, which is placed near the eyes. A compound microscope can give us up to 1500 times magnification. Do you know that the microbes are naturally colorless? So, when we look at them under the microscope, they are difficult to differentiate from the background objects and human cells. That is why we have to use various types of coloring dyes to make them appear colorful against the white background. This also helps us classify the bacteria based on their color, shapes, and sizes. Gram stain is the most commonly used dye to differentiate bacteria into 2 groups. Gram-positive bacteria appear purple and gram-negative bacteria appear pink after staining with gram stain. But there are some bacteria that cannot be colored easily with gram stain. So, we use fluorescent dyes for their identification. Once they take up the fluorophores, they can be easily identified under specialized fluorescent microscope as shiny things. This method is widely used to diagnose presence of tuberculosis bacteria in sputum samples. Other than the fluorescence, we can also use dark field microscopy to visualize microbes that are difficult to stain. Under this microscope, we can see illuminated live motile bacteria against the dark background. The process of coloring with dyes kills the bacteria. But if we want to examine them in their natural state without killing or staining them, we can do so by using a phase contrast microscope. It uses the phase shift in the transmitted light to produce contrast in the image. Another type of microscope is polarizing and interference microscope. It helps us visualize colorless and transparent objects, creating contrast with the background. All these were the examples of light microscopes that use light as a source of image formation. We have advanced microscopes that include electron microscopes and atomic force microscopes. Electron microscope uses a beam of accelerated electrons as a source of illumination. They can achieve a magnification of up to 1 million times. The electron microscopes have much higher resolving power than light microscopes and can reveal the structure of smaller objects like viruses. This table shows a comparison between the light and electron microscopes. The image obtained by electron microscopy is black and white, but colors can be added later, like coloring books, to create stunning images. This image shows a dry fracture of a cell where bacteria are busy growing. The atomic force microscope is also called scanning force microscope. It does not use lenses or beam irradiation. The information is gathered by feeling, or touching the surface with a mechanical probe, acting like a scanner with accurate and precise movements. This helps in studying the anatomy and physiology of microbes without killing them. So this was an introduction to various types of magnifying instruments we can use to peep into the world of microbes. If you liked the video, please press the like button and subscribe the channel. Don't forget to click on the bell icon to receive notification about new and informative videos. See you in the next video soon. Thank you for watching.